So, we've made it this far. Before I begin, I want to thank really not just the organizers to give me this opportunity to be here and talk about my work, but also for me the opportunity to be here with all of you. So I've heard of a lot of really great research here in these talks and in the posters and one-on-one conversations, and there's a lot more to be had in the program afterwards. And I think this is a really wonderful, unique opportunity and what I would like to call here physics Hogwarts. So I hope you all can join me in thanking the organizers to give us this special occasion here. And so of course here, final act. I promise to try to make it as entertaining as possible since it's been a long week. But before I do, I need to thank the wonderful people, instructors, and sponsors that make all of this possible. And so of course, none of this would have existed without all of these wonderful people. And if any of this resonates with you, especially for the undergraduates in the audience or anyone who knows an undergraduate that may resonate with the stuff that I'm about to present, you can consider applying to Texas. This is what our campus looks like, a very lovely, Italian-inspired architecture. And then to the left of the money shot is, of course, the brutalist physics building. We're together with math and astronomy. But of course, Austin's a wonderful place with a lot of nature. You go a few minutes out of downtown and you're in this wonderful green belt. And of course, all of this research and all of this spectacular nature, I'd like to acknowledge, takes place on land that once was inhabited by the indigenous people of this country, known to us today through the names of these tribes. So today, I'm going to talk about actin and myosin. Raise your hands if you've heard of actin and myosin before. A lot of hands have gone up. This is an active matter conference. And so for those that are not familiar, there may be a few of you. Actin's a rope. Myosin is a motor protein that likes to walk along this rope. And you get to see this walking action down here in this incredible fast AFM video from 14 years ago. And that's a myosin walking to the right. Every time the myosin takes a step, it burns an ATP, releasing chemical free energy, transducing that chemical free energy to mechanical work, motion, and that's ultimately what powers a lot of wonderful biological organization, flows, forces, motion, locomotion, cell shape changes, development, organismal shape changes, you name it. Literature is littered with actin and myosin mentioned. And it's not just at cellular scales, it's also at organismal scales where actin and myosin are present. Our muscles are chock full of these proteins. And that's what allows us to move around, and especially allows this one particular self-propelled active particle named, anyone who knows what it's called? Huh? Messi, yes, this is Lionel Messi. I have to show you a picture of Lionel Messi because my mom's Argentinian. I dare not show another soccer player. But here you're going to see, indeed, I guess a two-component active particle system interacting with a passive particle with a clear objective function, which is get ball and goal. And so, of course, there's all sorts of spectacular physics here. Look, not 1, not 2, not 3, not 4, not 5, not even the goalie can stop this goal. Growing up, I hated sports. I was that nerd in the corner. But now that I've studied physics long enough and I look at stuff like this, I love it. But it's not just organisms and humans that score goals. You see this kind of behavior also at the cellular scale. So I've stolen this video online from a neuroscience lab in the Netherlands, showing us a neuron. So here's its cell body, and it projects, all neurons project one single axon that connects to a whole bunch of other neurons. And in the developing brain, each neuron projects its axon, it grows, and it has to find a very specific goal, a very specific target. And if it doesn't find the right path and the right goal, then of course problems arise. In order to show you that this behavior, this goal, or goal scoring, is very similar on cellular scales and organismal scales, I've overlaid this video with audio from none other than Andres Cantor. Now I know some of you have seen this video, so you're not allowed to raise your hands, but for those of you that have not seen this video, raise your hands if you've heard of Andres Cantor. You're the first one I know to have gotten it.
Otra vez Chaco jugando para Zavala. El bullet. Balón filtrado, viene Chicharito. Deja el camino Nava, la tocó. Viene el remate. ¡Gol! ¡Gol!
Yes, that's 5 times Emmy Award-winning sports broadcaster. And yes, Mom, famous Argentinian, Andres Cantor, telling us that indeed there are similarities between cellular and organismal scales when it comes to mechanical behavior. And as a physicist, I find that fascinating, especially since these systems are driven ultimately by microscopic activity, micron scale chemomechanical activity. So how is it that this whole ensemble and plethora of little tiny microscopic events give us these slow variables, coherent biological mechanical behaviors, both at cellular and organismal scales? As a physicist in my PhD, this question already fascinated me, but it wasn't until my postdoc in mechanical engineering where I really, things really started clicking for me. At least this is the way I see things. That's when I started learning about robotics. What I've learned, robotics in one slide, is that first of all, the robot needs something to make it move, and that's here, this DC motor. You zap it and it spins. And then of course, you can't just, you have to turn it on and off, and that's what a control signal does. You regulate the voltage and you regulate how fast the thing spins. Furthermore, in order to get a good functioning robot, you need sensors. You need to know how fast this thing is spinning. You need to know, the state of your system. And when you have that information from your sensors about your actuator and the rest of your robot, then you can start doing cool stuff. You can develop a controller, use that information, write some software, and then issue new control signals, which then cause the actuator to spin left and right, and you can develop a control pattern that will guide your robot and keep it upright, make it do the thing you want it to. When I look at this as a physicist, I see that there's a constant flow of signals and information in robots that keeps the whole thing functioning. And then, of course, none of this happens in equilibrium. I mean, every robot needs a source of fuel to inject free energy into the system to sustain this flow of information that is always going on. And this is not just in robots. Cells do, in my view, the exact same thing. I mean, they also have actomyosin as actuators. There's tons of others. And then, of course, there's tons of different kinds of sensors. Here I'm showing you transmembrane stretch-activated ion channels, but you can imagine plenty more. And then, of course, there's, any systems biologists can tell you about the, headache-inducing networks of interacting proteins, just like interneurons in neuroscience, that then take these signals, process them somehow, in order to then, generate control signals, typically through phosphorylation and dephosphorylation, although there are other mechanisms too, that then turn the actuators on and off. And of course, all of this is sustained by chemical fuel burning. And so this is why I think control signals are really fascinating insight, not just from let's build something perspective, but to try to understand what's going on in biological and active systems perspective, which is why at APS, I, together with Aaron Teich from Wellesley College, who's also been interested in control theory, we've been organizing this wonderful focus session, graciously sponsored by DSoft and also DBio, about control strategies. This year we have a great lineup of invited speakers, including Danny Bassett, who has worked on neuroscience control strategies, and Luke Davis as well. Hopefully you'll find this as interesting as we do, and you may even consider submitting an abstract. So onward to tell you about the work that's going on in our lab. We indeed work with this actomyosin active gels. They consist of actin filaments forming this spaghetti-like meshwork, myosin motors, which exert contractile force dipoles inside, and then these fasten crosslinks, which introduce connections inside that are necessary for the contraction event I'm about to show you on the left. On the left, you'll see an actual experiment of a gel in a more or less pizza box-shaped geometry. You see that the scale bar is 1 millimeter. These things really are visible with the naked eye. They're about 2-3 millimeters in X and Y, and 1/10 of a millimeter in Z. So you can indeed picture a quasi-two-dimensional system. We just mix everything together. Actin filaments polymerize quickly on the scale of a minute or so. And then over time, the myosin motors cause contraction and cause the whole thing to shrink, as you'll see here on the left. And it's this contraction event, which mimics what's going on in actual cells. But notice there are no cells in here, nothing's alive. These are just purified, reconstituted proteins. So I think it's pretty cool. It's a nice active, minimal system that lacks the inherent complexity in real living systems. And then, of course, if you track the boundary of these gels and compute ultimately the strain as a function of time, you get curves that look like this. Over time, the strain develops and then at some point ceases, contractions over. And you can kind of see that here as the strains reach essentially a steady terminal value. They all do so with approximately 250-second time scale. It's a couple of minutes. And I'll also highlight here that the terminal strains here tends to be of around 1. And so I've been thinking a lot about this data because it seems so tantalizingly simple, so we might as well start with a very simple model. Imagine, I start, far more generally with like, stress balance and making assumptions that things are isotropic and so on. Ultimately, it'll all boil down to this nice, simple first-order differential equation where we have an active stress, pressure-like stress that's causing the gel to shrink. And then, of course, it's resisting that shrinking with some sort of bulk modulus and some sort of bulk viscosity. And then here's the strain. In order to solve this, it's just a first-order equation, and many of you can do it in your head. But I'm gonna walk a little through an alternative way that some people may not be familiar with. Raise your hands if you've seen Laplace transforms before. All right, good. More folks than I thought. I know in my physics education, I never encountered Laplace transforms. Fourier transforms, definitely, but not Laplace transforms. I think Laplace transforms here make more sense because otherwise, instead of s's, you have minus I omegas, and then everything would be imaginary because there's no oscillation in our system. And so when you introduce, Laplace transform all of this, you can then solve for or construct this parameter here.
Sorry, your left-hand side is missing the value of t0.
It's missing the value. Strain at 0 is 0. Yeah, but it would be there. You're right. Yeah. So I can construct this quantity, and then what I like about this quantity is this value here on the left. If you in stat mecha, you'd call this a response function. In control theory, you call this a transfer function. But it's essentially the bang for your buck. How much strain am I getting for the amount of active stress that the system is exerting? And well, you can solve for that. It's just a first order solution. For those that are not too familiar with control theory, there's a pictorial way of representing this. For anyone that's used LabVIEW and doesn't like it like me, because they weren't trained in using it, this is the foundation of LabVIEW, and it's just here's the transfer function or the response function. This is what you put in. This is sort of what's driving the system, and then this is the result. So there's sort of like implied causality here, whether or not there actually is there, but at least conceptually, the idea of input and output, I'm going to use later. All right, and so fine, we've got this response function, a response now to active stress, and then you can reverse Laplace transform, and it's just an exponential rise. It's a simple solution. And then, so now I'm going to ask what these terminal strains and relaxation or contraction timescales should be. And so I'm going to be a troublesome experimentalist. I'm going to take numbers from the literature to come up with, estimates. And I get numbers like this. I expect that the gel should strain a whole bunch very, very quickly. And that's not what we observe. Linear model over-predicts everything. And so something funny is going on. And well, it's really hard to dig deeper here. And this is something that we've struggled a lot, and we saw also with Aparna's work, is that this troublesome active stress. It's hard to measure. Sure, there's been some work done, estimating at least magnitudes of active stress, and I do estimate active stress through this Bendix paper, but getting a good experimental handle on this in active systems can be really, really difficult. And I already see a question coming up.
Yeah, so I guess this is not where you're going, but I see another issue, which is that you're assuming that there's sort of a fixed reference state, but this thing might very easily sort of plastically remodel. So if I turn off the active stress in the middle of my experiment, would it go back?
It won't.
In the model, it will, right?
It will not,
in reality, it won't.
Sorry, what?
In the model, it would, right? The model assumes that it would go back.
Correct. I have not included plasticity in this model, and so I'm ignoring that part. But it's true that there's plenty of, the system is completely plastic in real life.
But somehow that's not an issue for.
I haven't even figured this out yet, so I'm going to worry about step 2 later. I'm going to walk first before I run. Okay. But as an experimentalist, how can I try to get at some sort of response function? And so, well, here we have this viscoelastic response function or transfer function that ultimately these active stresses we know, and we see this over and over in the active matter field, that self-propulsion, active stresses ultimately come from energy consumption. But experimentally, energy consumption tends to be a quantity that's poorly characterized. But that's where it all starts. And so I'm wondering if instead maybe we can start including, at least in our experiments, mechanochemical response from that myosin motors that take in this free energy, produce some active stress, and that active stress turns into strain. And so I'll be working with this quantity little g, which is just an energy density that's consumed, chemical free energy density that's consumed. In the literature, sometimes you'll find it as like, sigma A or J is equal to zeta delta mu, so the change in chemical potential, but that needs to be made intensive for this to be the same. But either way, the idea is still the same. You can think of this J over G as a zeta. And then a question, of course, as an experimentalist is how do you measure this Gibbs free energy density? I'm not going to go into the details. This is sort of, definitely something that we did not develop, but actually got inspired last time I was here in 2019 for Cellular Energetics Conference. I learned that Zvonimir Dojic had implemented this much earlier, and he wasn't the first one to use this. This is a biochemical trick from long ago. But there are these two chemical reactions. If you want the details, we can talk later. That I include with the gel, or I should say my graduate student includes with the gel. Important thing is that there's this fluorescent molecule here, NADH, and it gets oxidized into NAD+, that is not fluorescent. And so as ATP is being hydrolyzed, NADH is a fluorescent molecule. NADH is being made not fluorescent anymore. And so now we can, we've introduced another unknown, but at least, we can start treading and trying to do some reasoning here. And so let's say the active stress is still constant throughout the duration of the experiment. Maybe that's true, maybe not. And let's also start simply and assume that this transfer function, this zeta, this J over G is linear and constant, and life's great. Then we would expect that the total amount of energy consumed would just, reach some sort of constant really quickly and end of story. And indeed, the initial expectation in the experiment would be, for example, here on the left, I'm going to show you another contraction event. Gel contracts to a little tiny square as before. And then, of course, this expectation here on the right is that a finite amount of blue would be made black and stop after contraction finishes. So the total amount of energy consumed would be somehow constant. And of course, that's not what we find. We find something different. We find that energy is getting consumed more and more and more and more, well past the 250 seconds it takes to contract. And so that was a surprise that we got this consumption after 250 seconds. And so what this means is that it's not the total cumulative energy that's constant, but, I'm not showing work here on slides, but we've done the work simulating the system with reaction diffusion and everything. And we're pretty confident that it's the rate of Gibbs free energy that's approximately constant to the best of our knowledge. And so that the total cumulative free energy scales with time. And so this means we kind of have to go back to the drawing board, taking a look at this picture here and asking, well, what's the minimal thing that we need to change in this picture in order to start being more consistent with experiment? And this is where my mind as an experimentalist started spiraling, because there are all these different kinds of mechanisms you can start tossing in there, and you can make your model very complicated. This is my current thinking, that there are nonlinear properties going on, that as you're contracting, the properties of the gel are also changing. So it's a nice simple linear model where we have active stress, we have bulk viscosity and bulk modulus and bulk viscosity. The active stress scales with the distance between motors. So as it contracts, the total number of motors is the same, but now they're in a smaller space, so you would expect the stress to increase. But then also, as your motors are increasing their stress, so is the bulk modulus increasing. And that's because you're fitting more and more cross links in the same amount of space. And then finally, the viscosity is also changing because the mesh size through which fluid is getting squeezed out is also changing. And so you can pause it, perhaps, as a first step. these sort of scaling relations with these exponents. Who knows what they could be, but at least maybe they're naive expectations. But then, of course, how can we start to test this? And of course, this is difficult to test because now we're introducing yet even more unknowns into our experiments, and so it seems like it's getting out of control already. And this is where we turn to nature for inspiration, and there's this, I'm going to show you this movie of what's called pulsed contraction. So we know that cells don't just contract everything all at once, but rather they contract a little bit here, a little bit there. Sometimes you'll find waves. But this movie, you'll see in red an upstream regulator of myosin, and you'll see that these little red dots, they form little clusters and then they just expand here, they'll form clusters there, sort of like a game of rugby. And whenever you see a cluster, that's where myosin is being more activated. And so it's not very common that you'll find the same contraction events that we see in our experiments inside cells. And so with this pulsed contraction, we mimic this in the lab, also with, I failed to use the word optogenetics, it's a cheaper trick than that. We just have a molecule called the caged ATP. It's a kind of ATP that doesn't work until you shine light on it. The protective cage comes off, and then the ATP becomes uncaged and available to myosin to perform the power stroke and this chemomechanical activity. And that's precisely what my student James does. Here you see a gel, and you'll see that he'll blast it with little pulses of UV light, and for every pulse, the pulse will be invisible, but you'll see the result of the pulse, and you'll see just little tiny contractions proceeding one after the other. And so indeed, you can plot the strain as a function of time. As a response, so you see the response to the initial pulse, the gel strains a little bit. Add more pulses, the gel continues to strain a little bit more. And then what I like about these plots is that we have a good sense of what each incremental strain is for each pulse, and how we can backtrack out how much energy was consumed for each pulse. And so with delta epsilon, delta G, we can do the following thing now. And this is the trick. If we deliver just tiny packets of energy to our system instead of just flooding through all at once, I argue that we're doing quasi-static driving of the system. It's being driven through phase space a lot more slowly. The strain rate is small, and we can start neglecting any nonlinearities in the mesh size. So already that starts to simplify things. And then, of course, we can now start with an initial assumption, and that is that, well, if the nonlinearities in J and K both rely on the shrinking of a distance, that scaling relation should be the same. So we could posit that A, alpha and beta should be the same, which then simplifies this as well, which leads us to the expectation that this transfer function, now this quasi-statically driven transfer function, should be constant. And so indeed, that's what we plot here. That should be our expectation if all those assumptions would be correct. And well, we find that that's not what we find. Instead, we find what appears to be a negative scaling of the transfer function with the free energy, leading us to say that indeed this transfer function itself is nonlinear. What I like about this result, in the world of rheology, it's kind of, as an active matter person, when I look at the world of rheology, it's great. You can measure stress and strain at the same time. And you can do all sorts of great nonlinear stuff and characterize the system, down to the bone. And here in active matter, often we typically only have the thing that we observe, and it's hard to get the thing that's driving it. It's hard to get that active stress. But at least what we can argue here is that we can get a good estimate of what this transfer function is, so we can get some sort of, modulus-like quantity or compliance-like quantity. And we have evidence that it is nonlinear. And that means that as the gel is contracting, that minus sign up there is telling us that as the gel contracts in our experiment, the next packet of energy yields even less strain than before, so it becomes increasingly difficult to contract. I can't tell you what exactly alpha and beta are, but at least this value provides a constraint on the two. And so when I think of contraction, I like to sort of zoom out a bit more, especially these nonlinear properties, and ask not just what contraction is mechanically, but what it is in terms of control processing and information processing. You can think of contraction also as a sort of transfer function, but then ultimately responding The energy ultimately comes from pulsed control signals coming from phosphorylation, dephosphorylation events. And then ultimately, contraction causes mechanosensors in the cell to activate, and so you get pulsed output signals. So I like to think of the actomyosin cytoskeleton not just as something mechanical, but also like a software component or a circuit component that interplays with all the intracellular signaling processes. And so I do ask, and I'd be curious to chat more over the next 2 weeks that I'm here, what the implications of this nonlinear contractility together with pulsed signaling is, especially if you compare with the steady signal case. Kind of like how in radio you have amplitude modulated and frequency modulated. Here we can compare amplitude modulated versus pulse width modulated information propagation. But I think maybe you know, this nonlinear contractility could provide energetically economical benefits, essentially, especially since delta epsilon over delta G is a sort of like a energy economy quantity. And then, of course, you know, how does this affect the transmission of mutual information? And I think all of this stuff fits in, especially when you then start wanting to quantify how actomyosin contractility fits with, ultimately, mechanochemical feedback processes inside cells. And so I've got 3 minutes left. I am going to, how about this? I am going to give the one slide pitch, and then I'm going to leave you all with a movie. I just can't bear to not talk about Jake's work. So Jake's been interested in muscle, which is also driven by actomyosin contractility. Did a heroic effort diving into the literature and looking at muscle nonlinear force generation, and you know, collated results from a whole bunch of studies. I'm not going to go into this nonlinearity parameter alpha. If you want to talk more about it later, I'd be happy to. But we find consistent with what people have been saying, at least informally in the literature over and over, is that there's a nonlinearity parameter in muscle that is not zero. So there's a nonlinearity in muscle. And so we started this project by asking whether there's something optimal to this nonlinearity, and we approached this optimality from the perspective of energy consumption and work production. And we built a robot to test this. And so you know, and also developed a model of actomyosin in muscle as well to compare with the robot and find similar behavior between energy consumption and work production. And I think this notion of energy consumption is very important to characterize when we want to describe how active systems toss signals around in a non-equilibrium fashion. But I think it's wise if I just finish up with this entertaining movie from the RoboCup. Raise your hands if you've seen the RoboCup. Ooh, just a few folks have. This is the RoboCup. So Texas Robotics, plus many other you know, robotics groups in the world, sends teams of robots to play soccer to see who wins. My colleague Peter Stone in computer science at Texas likes to say, he's proclaimed, by the year 2050, we're going to have robots that are just as good as human soccer players. Now, this is well past his retirement date, so who knows? But and this movie was pulled from the 2010s, so you tell me whether we'll make it there or not.
A long way to go. A long way to go.
Yay. So yeah, just as how Aparna says, let's get little tiny bags to do stuff. Let's see if we can get robots to do better than this. I like this one. So hopefully we can do better than this by 2050. Thank you all for your attention. Yeah, exactly. It's just the timing. They're getting better. They're getting better. Yes. Yeah. But you can imagine a lot of AIs there in the decision-making processes. Yeah.
Wonderful talk. Thanks a lot. We do have time for questions. Video.
Yeah, sorry, I have to come back to my plasticity point. So
yes,
in the movie
please.
that you showed where you pulsed the activity, right, that sort of clearly showed that after the pulse, it doesn't recoil basically at all.
Correct.
Right.
Correct.
So that's plastic, fine. But I was wondering, do you know in more detail what actually happens with the myosin while there's no light? So I guess the motors stay attached, the heads stay attached because they need the ATP to actually detach and sort of reset the cycle. So then they act essentially as passive crosslinkers.
Correct. It's in rigor.
Yeah, but so would there be a way to actually release the myosin?
So if I had so there are folks out there that have much fancier setups than I do, where they can, rather than control the ATP concentration, control the actual myosin activity rate.
Yeah.
Not for the myosins that we're working with that ultimately are responsible for contraction, but rather more the ones that toss filaments around, and also kinesin. We saw that earlier. Guillaume and others, work with these light-activated motors. And I'd love to have something like this for contractile skeletal myosins. But I don't know if they exist, and if they do, I certainly can't make them. I'd have to beg for them.
All right, thank you.
Anton?
I was very curious about your plot of the different samples, right? And where they came from in the body. Is this just a sample-to-sample variation, or is there something systematic about it?
Are you referring to this plot right here?
No, I think it was towards the very end where you have...
Oh, thank you. It was this plot right here,
Yeah,
yes.
exactly.
Okay. Oh gosh, yeah, so I didn't include more. So here are different muscle groups, and muscle groups are mainly taken from human data, measured from actual human muscle in experiment, but there are also some non-human muscles in here. And so the question is, what's causing this spread? So our model has a parameter that can relate this value of alpha to microscopic unbinding. But that's also not the only thing that contributes ultimately to the phenomenologically measured coarse grain value of alpha. But at least we can show this relation is enough to produce a nonlinearity. Now, the question of why some muscle groups are highly nonlinear and why some are almost linear or why some are in between, my take is that in muscle biology, folks have kind of moved away from this picture of looking at this Hill nonlinearity parameter, since the initial experiments were done under very specific conditions, and I think more elaborate models have come around, and so I think a lot of folks have not been too much interested in this alpha, but more recent work that we found also has now related alpha to this unbinding that I could talk about later. And I think there could be indeed some biological currency to trying to interpret these values.
Very interesting. Thank you.
Thank you.
Hi, great talk. You talk about how you find that the strain is decreasing as the muscle or as the gel contracts, but do you have any intuition of what's going on with the stress there that the gel's producing?
No, and I would love to have that intuition. And anyone who can measure forces should be able to make some great statements there. And I think dream experiments to really nail down these properties would be energy and stress and strain all at the same time. But that sounds like a tall order experimentally.
Okay.
Yeah, very interesting. So when you write down models for things like cells or cell junctions, you often end up with a Maxwell model, whereas what you have is something like Kelvin model. But so usually, and then people make iterations on this one with some extra loops and circuits and nonlinearities and so on. So where do you think all of this here fits within that paradigm? Should we be doing this at all, or?
So my understanding is that I've also seen Kelvin-Voigt-like models in other active gel studies that are looking at the actomyosin cytoskeleton. And if I remember right, Kelvin-Voigt is when they are in parallel and Maxwell's in series, or maybe I got that mixed up.
Kelvin is the one which stops, and Maxwell is the one which becomes viscous. That's how I remember.
Okay. To me, this worked out quite simply because the stress is added up linearly rather than having to do 1 over 1 over. But if you have a reason why we should go with Maxwell instead of Kelvin-Voigt, I'd be happy to hear why.
No, I don't. I'm just saying usually what you get is some horrible nonlinear combination of several of these, which might...
I see. So combining many linear components together to form an effective nonlinearity. I haven't thought of that. And if it helps simplify things, I'm all for it. I'm just looking for simplicity and raw reasoning here.
Yeah, I don't see I can offer that bit.
Yeah, well, but the idea is still good. We can chat more.
Yeah, I had a question about, so in the video with the, I think, NADH turning from blue to black, it seems like the activity is very localized, like the strain thing. So do you think a model, like a homogeneous model, is going to work in the end, or? So
this picture of energy to active stress being completely linear, I don't buy it in the system, because Fritjof was talking earlier about how myosins actively bind and unbind, and me intuitively, I feel some sort of unbinding mechanism highly belongs in the active gel model, but I don't have yet the experimental power to be able to rule out other mechanisms and include this one. I'm trying to go for the absolute simplest thing that I can, and I think the dominant mechanism here might be indeed this nonlinearity, but it doesn't entirely yet rule out these mechanisms. Now, what I like about this robot project is that indeed it did give me a chance to look at attaching and detaching, at least in the context of the robot, and hopefully some of this thinking will filter back into active gels. Yeah.
If I may ask one question, you saw when you were contracting this more and more, it was harder and harder to contract, right? I recently heard a talk by Martin Lenz, where he was saying that if you have very strong contraction, that you have contact regions between the filaments, and the number of these contact regions increases, just as in sheep wool or some other material, and that's the effect of the nonlinearity that you have in these systems.
And that should make the sign positive instead of negative, or?
Should make it harder to compress.
Because of the increased contacts of myosin and
of actin with itself.
Of actin with itself. Okay, yeah, then I'd be curious to see more, because it seems like it might be similar. It could be that this mechanism could account for the minus 0.3 that we observe. Yeah.
Can you measure the contacts? No?
That sounds hard, but maybe there is a way. I'd have to think about that. I'll scratch my cowboy hat.
Good. Please.
So when you apply the UV light to the caged ATP, you're permanently uncaging the ATP, even when the light is off, right? So did you calibrate those pulses such that you think that you are uncaging the same amount of ATP, which is subsequent?
No. We didn't do anything that fancy. But we do know that, so although the pulses are equally spaced in time and the pulse width duration is the same, or constant for each experiment, we do 4 different pulse widths. We do know that every pulse indeed releases less delta G each time. Yeah, and that we can determine for sure.
So in the first pulse, you have like a small amount of ATP released in the systems, and if you just waited a long time, would that fully contract, or would it get arrested in some intermediate?
No, it would just contract a little bit, and then the only way it could continue to contract is if we deliver more ATP.
So that's because the activity level is just so low that it's...
That's simply because there's no fuel for,
so is that ATP is getting used, so you don't have, do you have a regeneration?
No, so in these experiments we don't have regeneration. It's a finite pool of caged ATP. Every pulse stochastically unbinds a certain subpopulation of those caged ATPs, and because there's a finite pool of caged ATP, that fraction of, the fraction is the same, but the total unbound decreases for every step.
Thank you.
You're welcome.
Yeah, very interesting talk. So for this kind of filamentous gels, it is known that there is a continuous transition induced by strain, right? So as you give it more strain, there is a transition that people have measured critical exponents, etc. I wonder if you have tried to fit your data with that.
No, so we've definitely been thinking about that. In a way, one could interpret this negative slope, also potentially coming from some sort of stress or strain stiffening, which makes it even more difficult for the gel to contract. Curiously, so this strain stiffening occurs under extension or tension. And these gels are contracting, so you would think, oh, they're under compression, not tension, but the motors are still pulling, so the filaments that are bearing the contractile stresses are under tension. But then we think there might be a background network of, like, fluffy actin that's getting in the way maybe, but that's all speculative. But we do think that those filaments under tension may be forming some sort of percolative backbone that could ultimately result in some sort of strain stiffening nonlinearity. But we don't have yet the experimental data, given that it's hard to measure J, in order to conclusively say anything about that. But that'd be super interesting, yeah.
Yeah, thank you.
Thank you for the question.
Yeah, if I understood correctly, so first of all, I want to know which myosin motor are you using? Is it myosin 2?
Yes.
So is it in like polymerized bundles or?
Yes,
because the question is because the bundle form, it has like multiple, like hundreds of heads. And when you go into a very super contracted state, like it's binding to multiple actin sites all at the same time. So it's highly unlikely for it to disassociate from actin filament. And the second point is, like, you are measuring the strain over a very macroscopic length scale, but probably when you are in a super contracted state, you need to zoom in and see the local strain, local changes in the structure happening.
All right, so the first question, yes, we do, so we have thick filaments. There are tons of, I forget the precise number, but it's either tens or maybe about 100 myosin heads per myosin thick filament, which makes them highly precessive, which indeed makes them stick onto actin, and they can keep churning through, and we like that because that gives us contraction. If we add monovalent salts, for example, to make those thick filaments smaller, no more contraction. And you still get some fun stuff, and you're nodding your head, so maybe you've also found some fun stuff too. But if you want the most oomph you can get, you want thick filaments. And so we keep the ionic concentration low to promote thick filament formation. Now, the second question, remind me, was...
even I forgot what is this. So I think you have to locally see the strains, I guess. Yeah,
local strain, yes. Yeah. All right. Here's a tip. Here's something that I learned in my PhD. I was given the task of quantifying these active gels. So what does a plucky young PhD student do? Goes on the microscope with a 100x objective with a gel that's contracting over millimeter scales. And then I started zooming around trying to follow these things that are just flying around everywhere. And I wasn't getting anything quantitative. All I was just observing was things flying around. And so it was very difficult for me at the time, the stage I was then, to be able to get anything useful. And then the insight for me and it took me months, is to develop these pizza box shaped chambers using a very cheap low power objective so I could see the entire gel all at once and see how it contracts at macroscopic scales. And that was sort of like the sort of orthogonality vector and methodology that I developed, which allowed me to sort of get a bird's eye view, and I found that to be very helpful. I still find it helpful here, and we're seeing evidence for at least some sort of non-affine displacements, under certain scenarios. So I still think we can see some cool, interesting stuff at a bird's eye view. But I'm glad you bring this up because maybe this is warranting us to then start going back down to the microscale again.
And I have one more suggestion. I don't know whether it will work. So I think you can do this experiment without any additional cross-linkers. So then the extent of strain that you will get will be much higher because you are somehow just limiting the size of the final structure just due to the cross-linkers. My intuition working with these systems in my postdoc, it just tells me I don't work with these systems anymore. But if you just contract the actin network, I understand it's difficult to form an actin network without a cross-linker, but at reasonably higher density, if you just throw in myosin, you can make much smaller gunk of actin in the end. So you will have maybe much higher range to play with. Just an intuition. Yes.
Yeah, and there could be games we could play in order to tune that terminal strain. It would be pretty cool. Yeah.
Okay. Thanks.
Yeah, thank you.
One final question.
Thank you for your talk. So I guess when you activate the ATP, do you get any change in the Z position? Does it buckle or anything?
All of this was taken with non-confocal microscope that has no Z resolution. I have done experiments in the past where I have looked at Z, but those were, I can't remember now whether I actually have Z projection or Z trajectories as a function of time. I think others have looked at this though, but so far here we work under the assumption that whatever strain we observe in X and Y is the same in Z.
Thank you very much.
Thank you.
Great. So let's thank Rahul and Jose once more for the wonderful talks.
Just want to thank you all for coming and particularly for staying until the end. Although those who did not stay missed the soccer robot, so they really regret that, and we hope maybe in 10 years you can come back and show us if they're actually doing any better. And also thank all the people who ran the sessions that really kept us in time, and we actually managed to have a lot of questions, which was really, really wonderful. I know some of you will be here next week, which is great. So expect an email over the weekend with information on what's going to happen next week. But most importantly, I think we should all thank Daniel Sussman, who is really the one who organized this wonderful conference. And Anton, who is there somewhere, who will be organizing the program for the next 2.5 months. So I hope you enjoy the rest of the afternoon. Maybe go out for a walk, go to the lagoon, go to the ocean if you are, unless you have to go to the airport. But the airport is nice too. Have a good trip back. Bye.